Hi, my name is Lionel Raymond. I have several appointments. I work with Kaplan Medical, giving reviews in biochem, genetics, pharm, path, integrated cases, conceptual lectures, bunch of little topics. I also work in forensic toxicology. I'm affiliated with University of Miami in the Department of Pathology with a forensic toxicology laboratory. And I'm also a private consultant with a firm in New York City, which is called the Forensic Panel, where I peer review expert testimony with other colleagues. Chapters 8, 9, and 10 in our biochemistry book are really what I like to call the miscellaneous chapters of biochemistry. You just went through the big section of molecular biology that will be further reinforced when you work with medical genetics. And then you'll be working from chapter 11 onwards on metabolism from 11 to 18. In those middle chapters, we have many little stories. They're all board relevant. And one particular aspect of these stories is that they're very easily integrated to other disciplines, whether it would be physiopath, pharmacology, in some cases you'll see some microbiology. So we can find some real neat, interesting points in there. In chapter 8, we're going to work on amino acids, on proteins, and also on enzymes. And you'll pay particular attention in this chapter to enzyme kinetics and the graphics, being that we use these graphics a lot in pharmacology and in physiology. So as we start with amino acids, an idea about structure. First in words, if you call something amino acid, well, obviously you must mean that it has an amino group, and acid, it has a carboxyl group. The key point about the general structure of all amino acids is that the carboxyl group and the amino group are attached to the same carbon. It's actually what we call in chemistry the alpha carbon of the structure, being that this is where the major functional groups will be located. Look, let's try and invent it. We don't need to really be chemists to do that. We put a C right here. Let's imagine this is the alpha carbon. Let's put an amino group. You remember amino groups are NH2 or NH3+. It'll depend, of course, on the pH. We'll put a carboxyl group. Let's imagine then a carboxyl group like COH, or as you know, it could be CO minus. It depends on the pH. So that now I have literally an amino acid with the amino function and carboxyl function attached to the same carbon. Now, carbons always have 4 valences, 4 bonds. One of them will be occupied by an H, and the last one, importantly, is where we're going to find what we call the side chain, which I can call R for right now. This side chain is what will allow us to classify the amino acids. The amino acids are going to be classified at the very least in hydrophobic, they hate water, and hydrophilic, they love water. Imagine a protein having hydrophobic amino acids. Then these amino acids hating waters are more likely to be located where? Are you saying membranes? Because you would be absolutely correct. And that hydrophobic pocket could be an area that loves lipids and that allows a protein to hook itself in the membrane. Could be the cell membrane, but it could also be the inner mitochondrial membrane, maybe lysosomal membrane, endoplasmic reticulum, and so forth. Conversely, imagine you wanted a protein to live outside in your bloodstream. Probably you want it to be hydrophilic. So we can certainly classify the amino acids based on their side chains and their liking water or not. Well, rather than looking at the text right now, we'll go back to it in a second. Let's start looking at some of these structures. Do you recognize the amino acid structure first? Notice that every single of these chemical structures has an amino group, here labeled as an H3+, a carboxyl group, here labeled as a CO-, that hydrogen on the central carbon, and then the side chain. And as I look at the side chain, here we're going to look first at non-polar, it means hydrophobic, they don't like water, aliphatic side chain, meaning they're going to be carbons and hydrogen on the side chain. These hydrophobic amino acids first are represented by glycine. Glycine has the smallest of all side chain. If it has the smallest side chain, it's simply because it's an H for the side chain. With a small side chain, you've already seen a key point about glycine for your exam. Do you remember the structure of collagen in molecular biology? When you looked at collagen, glycine was found every third amino acid. And with such a small side chain, it allowed the collagen to have a very tight alpha helix. That tight helix could then be proportional to the tensile strength of this extracellular matrix protein. Glycine, otherwise, I would always remember, is also a neurotransmitter. It's an inhibitory neurotransmitter, particularly in the spinal cord. And you may remember that certain bugs, such as Clostridium tetani, can have some toxins that decrease glycine release. And without the glycine, without the inhibitory tone, what then tends to happen to information getting to the muscles of a patient who has tetanus? Yeah, indeed. Without the inhibitory function of glycine, now you get excessive tone of the muscle with opisthotonos, with that actual seizure type of rigidity that is associated with lack of glycine. So no glycine, know that it's the smallest amino acid. It is hydrophobic. It doesn't have any oxygen, any nitrogen in its side chain, so it doesn't like water. And as such, in a regular protein, might also be part of intramembrane portions of that protein. Other hydrophobic amino acids include alanine and valine, but also leucine, isoleucine, and proline. And if you know those, you know the hydrophobic amino acid that have aliphatic side chain. What stories can we do with the others? Look, if you want, I would actually highlight well valine, leucine, and isoleucine. Look at the side chains. It's what we call branched chain. And branched chain has one purpose on your step 1 exam. Imagine you have that little baby with all kind of neurological dysfunction, plenty of things really non-specific that's going wrong, nausea, vomiting, and so forth. And the key point in that little baby, body fluid, particularly urine, smell like maple syrup. Don't use it on a pancake. But the whole idea of that maple syrup is that maple syrup urine disease is associated with a buildup of branched chain amino acids because of an enzyme deficiency in their catabolism. So if I'm putting branched chain amino acid building up here, it would be in maple syrup urine disease. Imagine what your board could then tell you. They could ask you which of the following amino acid would be building up in the urine of this patient. Then your choices would literally be valine, leucine, or isoleucine. I'll give you another point that you'll see later in your metabolic section. Leucine will have the key point that it is strictly ketogenic. Now, strictly ketogenic literally means that in metabolism, the only thing we can do with leucine is make it a ketone body. Ketone bodies will be important forms of energy that tissues can use, particularly heart tissue, kidney tissue, and the brain during starvation. So little point to know. Otherwise, proline, if you wanted to know about proline, you see that it is actually a cycle that makes the side chain. So it cyclizes on itself. So this is an amino acid that tends to disrupt alpha helices, and if it disrupts alpha helices, it allows the actual protein to change shape. So this is your first group of hydrophobic amino acid. There's another group of hydrophobic amino acid to remember well, the aromatic amino acid. First, they're called aromatic because they actually have a smell. Doesn't mean they smell necessarily very good, but they certainly would have a smell to be recognizable to a chemist. They actually include phenylalanine, tyrosine, and tryptophan. These 3 amino acids are relevant for having a ring in their side chain. And when you have rings in the side chain, that's how you become aromatic, particularly if you have conjugation of one double bond, a single bond, a double bond, a single bond, a double bond, a single bond. So that's classic of aromatic structures in chemistry. Well, they're all relevant for making some key products. First, look at phenylalanine and tyrosine. Can you see the only difference between these 2 amino acids? The only difference is an OH group. So should I be able to go from phenylalanine to tyrosine in my metabolism? You bet. All I would need is an enzyme that we're going to call phenylalanine hydroxylase. And that phenylalanine hydroxylase, I know that all of you remember that it is the enzyme that is deficient in phenylketonuria, in PKU. Phenylalanine is actually, we'll see, an essential amino acid. So in people with PKU, you know that then tyrosine would itself become essential. So phenylalanine is a precursor of tyrosine. Now what do I do with tyrosine? Tyrosine is a major amino acid to be a precursor of a number of things. Tyrosine is actually a precursor of melanin. So everywhere you see tyrosine, you should be able to see some pigmentation of tissues. Tyrosine is also a precursor to make catecholamines, and catecholamines include dopamine, I'm going to write DA, norepinephrine, I'm going to write NE, and epinephrine. As you look at this then, if the same tyrosine can be used to make melanin and also be used to make compounds such as dopamine or norepinephrine, would you expect some color to the actual areas that have neurons building those neurotransmitters? Well, I'm sure you would. And what do we call the area that produces dopamine, for example, in the midbrain and degenerates in Parkinson's disease? We call it substantia nigra. And that black color comes from the melanin. You know, for norepinephrine, you'll see that there's an area also in the brainstem around the reticular activating system that is called locus coeruleus. And coeruleus means blue, and that dark blue color comes again from melanin. As a matter of fact, when I'll have degenerative disorders such as Parkinson's and I destroy the dopaminergic neuron, what do you think a pathology question would ask you about the gross aspect of substantia nigra? Well, it would be depigmented, and depigmentation would be expected every time you destroy places that have tyrosine. Another point that we'll know about this is that as you go to melanin, this is done by a key enzyme that's called tyrosinase. And tyrosinase or tyrosine oxidase, which actually requires copper as a cofactor, is the enzyme that is deficient in albinism. So associate tyrosine with both catecholamine synthesis, that's dopamine, epinephrine, and also with melanin type of production. Tyrosine, finally, I would always remember my physiology. What do I do if I have two tyrosine residues that are adjacent to one another in thyroglobulin protein in the colloid of my thyroid? Well, those tyrosine, indeed, I can iodinate them. I can put some iodine, and as I put iodine with the enzyme peroxidase in the gland of the thyroid, I prepare my iodotyrosine to then further couple in MITs and DITs and eventually T3 and T4 my actual thyroid hormone. So tyrosine is also quite relevant for thyroid function in endocrine physiopaths. How about tryptophan? Tryptophan has several major products from it. If you want a neurotransmitter, do you remember which one we have? It's the one we need every now and then when we don't feel too good. You know, maybe we'll take fluoxetine for it. It is serotonin, 5-HT. Tryptophan is the precursor of serotonin. It's not only a precursor of serotonin, it's also a small source, but a necessary source of niacin, so that their niacin deficiency could actually come from tryptophan deficiency, such as we can see in what we call Hartnup disease, where there's a problem with levels of tryptophan. Well, we'll talk more about this when we review vitamins together later on. Tryptophan is also quite relevant for making not melanin, but melatonin. And melatonin in the pineal gland is important for circadian rhythm. Tryptophan, another important aromatic and yet hydrophobic amino acid used for a number of byproducts. Once you have all this ready, the only thing I would try and make you remember is out of your molecular biology. What was the primary signal in order to push proteins outside of the cytoplasm so that they would be placed in membranes, outside the cell, or potentially in the actual lysosomes? Do you remember the N-terminal hydrophobic signal sequence? I'm just going to call it here N-terminal signal sequence. Well, a signal sequence was when the first few codons of exon 1 of a gene would encode for hydrophobic amino acid. You now know how to recognize if the beginning of a gene would encode an N-terminal signal hydrophobic sequence. You see if it has lots of codons for tryptophan, tyrosine, phenylalanine, but also for proline, isoleucine, leucine, valine, alanine or glycine. If there's a lot of codons for these amino acids, you can be sure that this is a protein that will be targeted because its N-terminus will not like the cytoplasm. It will now want to be hooked on the endoplasmic reticulum for targeting to occur. How about being hydrophilic? We have some text there also. I'll show you the text at the end. Let's go to the actual tables. And as we look at the table, the first group of hydrophilic amino acid are very relevant from the standpoint that they have some charges on their side chain. What is common to all these amino acids here is that they have a positively charged side chain. That positive charge came from an extra amino group. This extra amino group is found in lysine, arginine, and histidine. Now for lysine and arginine, I know you already know something also from your molecular biology. What was necessary for proteins to interact with DNA? And recall, DNA has negatively charged phosphate groups. Well, it was important for these proteins to have positive charges to bind well to the DNA, to package it. And histones were a great example of proteins that were rich in lysine and arginine. Well, thanks to this, they could have those positive charges required to interact with DNA and allow tight packaging. And let's not forget, it's thanks to that tight packaging that we protect our nucleic acids from nuclease digestion so that we can preserve the genetic information. Other points about it, I would know that arginine is the precursor of nitric oxide, NO, which is required to signal information to smooth muscle cells and allow vasodilation. We'll review signal transduction together in a later chapter. And as for histidine, we always love to remember that it's the precursor of the major inflammatory mediator, histamine, that is not only used in acute inflammatory responses, but also in type 1 hypersensitivities. Recall that IgE-mediated mast cell degranulation causes this powerful vasodilator to be released, histamine. Histamine is also relevant for its role in peptic ulcer disease, as you probably remember. Well, these positively charged R group, because of being charged, they're hydrophilic. They love water. Other charged amino acid will potentially involve the negatively charged side chains. And negatively charged side chains are those of aspartate and glutamate. You notice that the negative charge is because of an extra carboxyl group. Aspartate and glutamate are also pretty relevant because they are excitatory amino acids. As such, they are found as neurotransmitters, particularly in the central nervous system. For instance, do you remember a drug such as felbamate or ketamine? These are two drugs that actually block glutamate receptors. So a role to glutamate is certainly beyond that of an amino acid of a protein to make it negatively charged. It's also a neurotransmitter in and of itself. The last group of hydrophilic amino acid doesn't have any positive or negative charges on the side chain. And you can look at them two by two. I always like to lump together serine and threonine because they each have that OH group in the side chain. And having an OH group in the side chain, it will be a site for O-glycosylation. If you recall in your molecular biology, this is a post-translational modification that occurs in the Golgi and that confers more water solubility but also some immunogenicity to proteins. Putting a sugar pattern on a protein ensures that it's either going to be recognized appropriately as self or non-self. So remember serine and threonine for O-glycosylation. I would also remember it for O-phosphorylation. And a number of kinases, which are enzymes that put phosphate on proteins, will do so by phosphorylating the OHs of serine and threonine. Then I would look at the next two together, and they are cysteine and methionine. What is important about cysteine and methionine is the fact that they have sulfur in the side chain. Notice that cysteine has SH groups. SH groups are relevant to make disulfide bonds. And disulfide bonds are changing the shape of proteins. These disulfide bonds might be responsible for the appropriate shape for function. For instance, you'll study in physiology the disulfide bonds of the hormone insulin. And this will always require two cysteine to make that disulfide bond, which is like this, SS. Now that's pure chemistry still, but if you want to have a nice story with it, we use also the cysteine as part of glutathione. And glutathione is known as GSH and is used to pick up free radical. So it's used to detoxify free radicals. Free radicals are molecules that have free electrons and that can cause tremendous damage to other structures in our cells. Because glutathione comes in finite stores, when we have issues with degenerative disorders associated with free radicals, we run out of glutathione. So guess what we're going to use as a drug to mop up the free radical in our patients? Well, we'll use an analog of cysteine, and the analog is going to be called N-acetylcysteine. Its job will be literally to bind to free radicals. Let me give you some ideas of when we use N-acetylcysteine. I'm sure you remember N-acetylcysteine is used in the liver toxicity of which drug intoxication? If you're thinking acetaminophen's hepatotoxicity, it's perfect. It's an antidote to acetaminophen's hepatotoxicity. N-acetylcysteine is also used in cystic fibrosis patients. It's used to fluidify the mucus so that they can cough and expectorate all that junk out and suffer less respiratory infection. And the final spot is a very recent place where I've heard of board questions in pharm, which is the use of N-acetylcysteine to treat hemorrhagic cystitis from cyclophosphamide. Cyclophosphamide being an anti-cancer drug, whereas Messner protects us from the hemorrhagic cystitis. N-acetylcysteine will treat it if it has already occurred. So three places for that cysteine derivative. The fact that it's going to be used in acetaminophen toxicity, it's going to be used in cystic fibrosis to disrupt mucus, and very much it can be used in the hemorrhagic cystitis of cyclophosphamide. All this because of these SH groups that can make disulfide bonds with other proteins and also pick up free radicals. Well, if you know all this for cysteine, I can't imagine where else it would fit on a board question right now. How about methionine? Methionine, look at the name. It has thio for the sulfur and it has met in front of it because it has a methyl group. The key point about methionine, it's its use as a methyl donor. As a methyl donor in your metabolic section of Biochem, you'll review it as SAM. Or SAM is the way we call it. It stands for S-adenosyl methionine. And this methionine is going to be used to methylate some very important thing. For example, do you remember the methylguanine cap of your messenger RNAs at the 5' end? Well, that methylguanine, the methyl came from a methionine. It came from SAM. Another big methylation, when we make epinephrine out of norepinephrine, it will also require this SAM. So two sulfur-containing amino acids that are hydrophilic, cysteine and methionine. The last two amino acids on this slide are the amino acids asparagine and glutamine. And as you look at asparagine and glutamine, you're looking at amino acids that have an extra amino group in the side chain. Now, unlike lysine and arginine, they do not have any charges. Asparagine and glutamine are a spot where we can do N-glycosylation. And N-glycosylation is going to be another of these post or co-translational modification. Those tend to occur in the endoplasmic reticulum. And if you remember your molecular biology, it actually requires dolicol phosphate in order to happen. Dolicol phosphate being an intermediate of cholesterol. So now you know where you can put sugars, on the oxygens of serine and threonine or on the nitrogens of glutamine and asparagine. Let me show you that a lot of these points were already in your text. In your text, notice how it reminds us that Phenylalanine and tyrosine are precursors of all catecholamines. Tryptophan is a precursor of serotonin and niacin. Valine, leucine, isoleucine are the branch-chain amino acid that build up in maple syrup urine disease. Proline disrupts normal secondary structures. As far as the hydrophilic amino acid, serine and threonines are for O-linked glycosylation, a modification occurring on proteins in the Golgi. Asparagine, I added glutamine, are done for N-linked glycosylation. It occurs in the ER. That's not your emergency room, but your endoplasmic reticulum. Cysteine can make disulfide bond with the SH group, changing the shape of protein. And methionine is part of SAM, a methyl donor in biochemical pathway. All we've merely done is add on even further information that you probably recognize has been board tested. And unfortunately for us in that section, notice how everything is more or less straight recall. What do I do with the structures? Do you need to memorize the structures? What you need to memorize, my friends, is literally how do you recognize it's an amino acid? So you see the amino and acid group on the same carbon, and then just focus on the point in the side chain that gave you the question. Does it have a positive charge? Does it have a negative charge? Does it have an OH? Does it have an SH? And so forth. If you know what to look for, you don't need to memorize the structure, you need to recognize it. And then you imagine the question making you match the statement with the structure on your computer screen. If you look in your syllabus, you actually have a direct application of what we've done with the hemoglobinopathy box that describes to you a clinical application of knowing the charges of amino acid. Recall for that matter that the negatively charged amino acids are glutamate and aspartate. How about the positively charged one? Lysine, arginine, and histidine. Now let me take a sheet of paper and reproduce. The idea of electrophoresis is literally to have a gel, and on that gel to place proteins and run them in an electrical field. Here, let's imagine I'm putting my protein sample in the middle of that gel. As I'm putting the protein sample in the middle of the gel, I'm going to put two electrodes, maybe the positive on the left, the negative on the right, and then based on the charge of the protein, it's going to have to migrate through that gel. For instance, if the protein has negative charges, then it would be attracted towards the positive electrode. Negative charges would be associated with having lots of glutamate, having of course some aspartate. Contrast, if the protein has lots of lysine or arginine, then thanks to lysine or arginine, or potentially also histidine, then the positive charges would move that protein towards the negative electrode. So imagine a situation such as hemoglobinopathies, and amongst the pathologies of hemoglobin, think further about sickle cell anemia, a very famous hemolytic anemia associated with not only hemolysis but also vaso-occlusive crisis from those sickle cells, causing ischemic damage, potential infarction of tissue, such as the autosplenectomy that is so classic in such patients. Well, in sickle cell anemia, the point mutation is actually changing a glutamate at position 6 into a valine. And if that's the case, you're going from a negatively charged amino acid to valine, which has no charge. No charge means it's less negative, or if you want, it's more positive. If that's the case, then if my protein in the middle used to be HbA, HbA with its glutamate would have migrated towards the positive electrode. HbS for sickle cell anemia without the glutamate would probably stay much more towards the middle of the gel. Conversely, there is another disease that is called HbC disease. HbC disease is a rarer hemoglobinopathy, but if a sickle cell anemia patient had it, it would dilute the sickle cell protein and as such decrease the chances of sickling. In this one, the glutamate at position 6, which used to be negative, has become a lysine residue. And lysine, as you know, is positively charged. So this time, it is as if I got two positive charges extra. Since I lost a minus, it's as if I became more positive, and I further added an extra positive charge. So where would you put on my diagram HBC? HBC having lysine, HBC should be located closer to the negative electrode. So now if you look in your book, you see that we can differentiate the various hemoglobins of a patient based on their location on a gel. All we need is, of course, to run controls of this hemoglobin in parallel to the hemoglobin of the patient and then see where does the patient's hemoglobin migrate. If it migrates next to the HbA, then you know it's a normal hemoglobin. On the other hand, in the diagram you have, if it migrates next to HbC, then you know the patient has a hemolytic anemia, which is known as HbC disease.